Welcome to CC Worldwide Voices, and a very special welcome to Dr. Elaine Ingham. Dr. Elaine is widely recognized as the world's foremost soil biologist. For over 4 decades, she has been advancing our knowledge about the soil food web and is passionate about empowering people to bring the soils in their communities back to life. She has held many impressive roles, including the first president of the Soil Ecology Society, the program director for the Ecological Society of America, chief scientist at the Rodale Institute, and she started Soil Food Web Inc. 26 years ago. She currently serves as the Soil Food Web School's founder, principal ambassador, and science and research advisor. Her pioneering soil food web approach has already revitalized over 5 million acres of farmland around the globe. Very impressive. And it offers us a beacon of hope for sustainable agriculture and environmental conservation. Thank you again for joining us today, Dr. Elaine, and I'll hand over to you.
Okay, thank you very much for that introduction. Hope I can live up to it. So it would suggest to me that I might try a name put on here of restoring the world's growth, one garden at a time. And to me, that is, we don't do different things in different places because it's the size. We don't do that. We're going to look at what is present. We're going to figure out what that area with this particular problem or needing some help in some particular aspect, and try to make that function again. So if it's a pot that's 6 inches by 6 inches, we're going to be using the same technology to extract the organisms from the compost, to have made the compost in the first place where it didn't have any toxic chemicals present in that compost. It didn't have any disease-causing organisms, or not detectable, or very low numbers, just like your drinking water coming out of your faucet. So that might work just fine for 6 inch by 6 inch, but it's going to work fine for a 600 feet square. You could use exactly the same management practices. You're just going to have to use larger equipment. You may have to train a couple of people. When we get it to even higher levels, working with 2,000 square miles, it's the same kind of approach to fixing the problems. When there's been a time or two where we've had 10,000 acres of land that we're working with, all supposedly banana. Well, what we're doing over here is quite a bit different than what we're doing over here because of the topography, because of the different things that are, you've got to pay attention to the things that are not biological, but you also have to look at what the organisms are doing. So restoring the world's soil one garden at a time, I kind of feel like it's the other way around. We're fixing the soil because we have a knowledge of what's going to happen as we use the approach that we've used before. So, I'm going to try and look at whether we've got a small area or larger or larger or larger. But I do have to admit that we've looked at more than 5 million places on the planet that we've worked at. We probably have doubled, if not tripled, that much over the course of the last couple of years. So when we walk into a place that somebody has hired us, somebody has said, can you fix this? What's wrong? What do we do? We try to make it very clear that this is not a one size fits all, because there's so many different things we have to keep track of when we're using a biological approach. We're keeping the good guys and we are getting rid of the bad guys. That's highly scientific verbiage, don't you think? The bad guys and the good guys. And it does strike some people with a attitude about, well, she can't be she's not, doesn't really know things because bad. What is this? Bad organisms and good organisms? Well, it's a whole lot easier than trying to keep track of the 2,000 or 3,000 species of bacteria and or fungi in any place that you go to on this planet. You should, if it's even pretending to be healthy, you need to have that much diversity. And of course, this group of bacteria and or fungi or protozoa or nematodes or microarthropods or earth all of the different organisms that can be present in the soil, they all have differing things that the plant needs from those organisms doing their jobs. So I want to go through these jobs that the plants have. How do they get the good stuff from there to over here? They don't just, sugar, glucose, fructose, whatever, any particular thing. They don't get up and walk over there for the convenience of the plant. They've got to be brought along. They've got to be carried by something. There has to be, say, with water. That moves things. The organisms, as they walk by, are going to pick up all kinds of things on the outside of their body. And what effect does that have? We need to start understanding these things, but I think of it as almost like, the pyramid, where we've got the top of the pyramid with the kingdom, and then we've got the next layer and the next layer and the next layer down to the species level with bacteria, the fungi, protozoa, all of those things. So how does that all fit together? I'll probably answer that question in a little bit. So if I could have the next slide, or the first slide. I haven't even gotten this far, and look at this. I'm already off. Okay, so this always reminds me about this field. It belongs to Oregon State University, back when I was a professor at Oregon State University. This is where I did a few of my testing. And here we're looking at that whole field, and you can see where the driving lanes are, and we're right near maybe 10 feet from the edge of this acreage. And I like to remind myself exactly what I was just talking about. Let's take a look at what's going on in this field. What do you have to pay attention to? What are you going to have to make certain that you're catching the important difference as management is performed on these systems? So first of all, you can see the soil food web in all of its wonderful glory, slightly etched over this level. That's me with my fingies getting dirty in the soil. And one of the things I always want to bring to people's understanding is that when we take samples and put it in a plastic bag or jar or something like that, we don't put in ice. We don't put in something that has such a huge difference in temperature, because temperature is a huge selector of what's going on in that soil, in that system that we're looking at behind myself in this particular instance. So we want to make certain that we're interpreting the data that's right in front of our eyes. We don't want to be taking what was normally at a, maybe a 65 degrees, and you put it next to a block of ice. You're starting to change the biology in that ice, in that sample, because the ice is changing the conditions for the microorganisms. How rapidly do the organisms respond, or the fungi, or the protozoa, or the nematodes, or microarthropods, or the earthworms, or the whole list again. And it can happen within a couple of seconds that you're starting to see some of the organisms going into a spore form, or other organisms are waking up and starting to do something. So you just messed up our system of trying to understand what's going on in this field, because you're already changing things due to an outside parameter. So if you want to understand what the organisms were doing at noon o'clock, you better have a container that maintains the temperature after, closes off the temperature that might get colder, it might get hotter. We don't want that happening inside the little container holding area. And so you start thinking of all of those things. We'll go through a whole list in a little bit of things you've got to think of, and you want to keep them the same or as similar as possible, or you are causing imperfect data to be collected. So we want to take a walk. As I would stand up and turn around and start looking at this field, you can see that over on the left-hand side, there's a definite kind of a tan yellow color over on the others on that left-hand side. And when you put your hand in there and you feel the structure of that, well, it's not soil anymore, because all of the aggregates that used to be in that soil before somebody went in and started plowing, started tillage, and about 50% of all of the organisms in that soil were destroyed through the management practices used to get nice, smooth samples where everything is homogeneous. That's not telling us what's really going on here. It's telling us what would happen if somebody came along with a great big, something that ground everything in that soil up. I love it when my brain goes, bleh. What was the word you were looking for, Elaine? I don't know. So please excuse me. I apologize for those. Yep, I'm over 70, so it happens to us every once in a while. So looking over on that left-hand side, is there something that you can looking at that, is there something that tells you, are you the same as what this area would be if you had not tilled it up, in so many words? So we look at that whole left-hand side. You can see that there are a few small, well, they're not really aggregates, it's compaction that's formed those little divots. But over here on the right-hand side, look at the fact that you don't have nice, smooth land, that there was these dimples, there were compacted areas that as the tillage equipment went through this area, it broke down the first time they went over the ground, you can see that there was quite a few of these big rocky, well, you'd think they were rock just by looking at them, but no, indeed, if you go over this another 2 times with your tillage equipment, it turns into what you see on the left-hand side. The left hand side has already been tilled 3 times. Well, if we lose 50% of the organisms in that soil every time you run them through a tillage equipment, how much of that original biology are going to be in that soil? Is that soil? It's been tilled to death because you've lost 50% on your first pass around, another 50% on your second go around, and another 50%, and then you decided to leave it alone. This is not soil. And that's what we keep doing our tests in when you look at the chemical world. They destroy the biology, which are the very things you must have in order to understand how we're going to make it through the next 100 years, 200 years, or maybe we only got 10 days. We know we're going to have trouble unless we can get people using non-chemical materials to grow all of their food. And when we look at the cost of doing the chemical approach, it's much less damaging and it's much less expensive to use a biological approach. We just need to understand that system and work on it. We can, as we've been working with places around the world, we have shown that we can at times get the biology back into the soil and really help it, really improve it, build that organic matter layer within the first growing season. And the next year, what we can see, what we have seen, is an increase of 300% in yield with the maximum potential nutrients without without any use of equipment going back and forth across the field and compacting on one side and breaking it open on the other side. So what exactly do we have to do? What are the things we need to know? Next slide, please. So just a little bit of, put the BA, the MS, the PhD on there, where you want to get hold of me. So if you don't have my email address, ERI, that's my initials, at soilfoodwebinc.com. So good place to get hold of me. Next slide is looking at the second in command, Carla, Portugal. She is from Brazil. She's not with me this time around because she's just returned from a long stint in Portugal, and so not here. Next slide. The 4 of us in the group, and so if you call us at these not call you, but contact you through the Soil Food Web Inc. You'll catch one of us. So we're just in the process of expanding outwards, and things are going pretty well. Next slide. So here's the food web. So when we're looking at and trying to understand all of these organisms, we want to have a full accounting of what's going on in these soils so that we can determine the best management practices to utilize when we're trying to get maximum. Well, you're going to have to fill in the word. What are you trying to achieve? You can't just walk into my office and say, we want you to fix this. Well, how much area are we talking about? What's the extremes? What are some of the problems you've experienced in the past? Is it typically a root feeding nematode problem, or is it typically because you don't have enough bacterial feeding nematodes, or the fungi are only in particular type of fungi, and all of the mycorrhizal species are just not there? Which one is causing the problem? Which one is preventing that plant, whatever you happen to be growing? What prevents that plant from growing as rapidly as it should? And so we need to be asking questions. What has been happening in the past? What has happened when you've seen some of these interactions between some of these organisms? So let's take the soil food web from the top, which is, of course, sunshine. Sunshine, going to take that photosynthesis and convert it into a whole bunch of sugars and carbohydrates and things like that. And of course, then some of that is sent down into the root system. Now, often people will kind of object to what I just said because, it's the photosynthesis. That's really the only thing that we should be thinking about. Well, there's a lot of carbon that is put into a saved form on the inside of that fungal tube. The fungus will take those carbon compounds and lay them down on the inside of that hypha that you're looking at. And thus you are making certain that you're keeping more carbon than anybody ever thought the fungi could do that thing. When we're looking at the mycorrhizal fungi, they too put the nutrients down, mostly carbon, but there has to be some of those other nutrients in there so that the plant gets everything it gets what it wants. So nutrients going down into the root system from above and the roots moving out. Now, I remember when I was a kid being told that the root systems of trees or shrubs or most vegetables, certainly absolutely the case if we're dealing with grasses that are early successional grasses, they're not going to put their root system down more than maybe 5 centimeters. That would be a deep amount of material going into the soil. And so, they stop at about 3 to 5 centimeters, and that's then they're going to go sideways. Well, if you start digging in here, and we have some fun with people who come and look at what we're doing quite often. We dig down on the sides. We go out at least to the drip line and we dig down. And okay, you're not hitting any roots this way, or very, very, very few. Let's go halfway between the drip line and the stem of the plant. And what we do is, as we start digging, is there's a lot of roots out here. And they don't stop and go sideways at 5 centimeters. Those root systems keep going down and down and down if they can. Think about the first picture I showed you of the field in Oregon, where we dug down next to those roots and next to the root space, and we just had massive amounts of roots, very thick, very compacted, just having the greatest fun. I see, I'm now noticing the hand. Am I supposed to do something with the hand? Nope, just keep going. So the how deep, and I love the fact that I think it's at the Iowa State University in the soils lab, the soils library. They've had a little plant, a shrub that they went out into the prairie looking pretty, wasn't, didn't look too happy, really dry season at the time this was done. And then they started digging down, and the roots on that plant, how far down something, when you look at my hands, you can see the height of the shrub, about a square worth of photosynthetic material. And they started digging down into the soil in order to put this shrub up on next to the door of one of the soils library. And they dug, and they dug, and they dug, and they dug, and they kept running into more roots. How far down did the roots on that shrub actually go? Way beyond what anybody at the time had noted. So here we are down at, well, give yourself a, come try, see who wins, Sorry, I forgot all my materials for making up a desirable guess. But just think of it in your mind now. How far down did the roots of that shrub growing on the Great Plains of the United States, how deep did those root systems go? Be honest. It goes down 25 feet. How much do we actually know about how roots grow? And it kind of worries me all the time when these things we just believed because we were told so. How can you trust some of the principles of what we've put out there? I think we've got a lot of thinking to do, and then we need to get out there and do the science. So our root systems are bacteria, are fungi, growing on exudates being produced by those plants, releasing those nutrients. And so the bacteria and fungi will grow faster, grow in a more healthy fashion, and perform their job that they're supposed to be doing. So they are decomposers at this level. There's Chrissy said, when transplanting, oh, it went away. So yeah, look at your, I can't find it. Okay, so go and find what Chrissy said because she was corroborating the information I was telling you. So certainly possible for those root systems to go down 24 feet. When we start looking at Douglas fir trees, how far down does Douglas fir put its root system? Well, and you better give those plants the ability to um, um, have the mycorrhizal fungal hyphae be included in what is just normal fungal biomass going along. So the bacteria and the fungi here are basically decomposers. So look down at the bottom of the page here. Second trophic level, these are decomposers, mutualists. There are pathogens in here. And if you do not protect your plant by getting the biology covering all of those surfaces, you're not going to have a healthy system. Parasites, also same problem. Root feeders, we don't want any of those. And if we know what we're doing, we are in every instance be able to get rid of those bad guys. Yeah, my highly technical scientific language. In many of these systems, for example, bacteria and fungi will actually take up nutrients from your plant and then basically go and follow the instructions that that plant gave to the microorganism. So the bacteria goes out, gets into the soil structure, and starts using its enzymes to pull out exactly the nutrient that the plant, or mix of nutrients, that the plant requires. And the bacteria and fungi then go back to the root system of that plant, and they remain there until the root feeding, not root feeding, until those things that eat the fungi and eat the bacteria. So third trophic level, decomposers, mutualists, and I'm sure from the point of view of the bacteria and the fungi with all those wonderful foods inside there, they kind of don't like what happens where the fungi and the bacteria are eaten. And because the nutrient content of those microorganisms is so much higher than what is inside the so much higher than the other sources of foods for those microorganisms, that those nutrients will be released by the protozoa and by the nematodes because it's just too much good stuff. And so they release it. And that's what the plant's going to take up. It gets its part of those nutrients. And so the plant moves on, telling the bacteria and the fungi that what it needs now is something different. And go find me that some of the difference. When you come back, I'll have the predators waiting here so that the bacteria and fungi will be eaten and the excess nutrients will be released into the soil. Now, it turns out that this is not just something that bacteria and fungi do. If any of you know the work by James, I'm not going to get his last name this time around, but at the University of in North England, New England, sorry, the plant is putting out exudates, attracting the microorganisms into that root system. And as soon as there's enough of those bacteria and fungi, maybe even a protozoan and a nematode in some cases, when inside, the plant will then close all of the access doors and release highly oxidated materials, which will cause the cell walls on the bacteria and the fungi to wear away. And so a huge amount of those nutrients come right out of the bacteria and the fungi. Well, the bacteria and fungi aren't having a lot of fun at this party anymore. So they're moving up along the inside of the root tip of that plant, and just so happens that there's a break in the root hair and the root part, and so all of these organisms can escape out of that small space, because bacteria, they aren't very big. Fungi squeeze their system and escape. And so now they've got all this great nutrient in the area around the root system of the plant, and they go on down to the tip of that root and repeat it all over again. So, we have some of our favorite, it's kind of fun to figure out new names for these guys, because it's amazing what goes on around that root system in ways we never suspected when we were killing everything that was in that soil. So let's, microarthropods, the nematodes, protozoa, they are what eat the lower level below them, the second trophic level, mostly consumed by third. And of course, Anytime there's a discrepancy between the nutrient or phosphorus or potassium or manganese or any of those nutrients, those excess nutrients are going to get dumped out into the soil, and that means someone downstairs or run around down there in the soil would be utilizing those materials, and diversity is what's really important. At all times, there's something to be fed on. So the nutrient cycling processes are allowed to be maintained. Your plant wants to have those nutrients. And so building that structural integrity is exactly what you want to do. Next slide, please. When we look at, for me, the pictures are right on top of is there any way for me to turn them off? No. Okay. I'm not going to worry about it. I can remember, I think. So now this is one of those things in understanding what's going on in the root system of the plant. People like to talk about dirt and soil as being one and the same. So when I was a graduate student at Colorado State University, working on my PhD with Dr. Donald Klein, it was one of those questions what's the difference? Dirt is composed of mainly mineral nutrients mixed in with the material sand, silt, and clay. And the percent of sand, silt, and clay varies, but ultimately the recipe is what is the exact mixture of all of these. And what you'll notice right off the bat is there are no organic materials. So this is what's coming from the bacteria and fungi going up to a clay, a silt, a sand particle, growing on the surface of the boulders and the rocks and the gravels outside, converting some of the salts and the solids then into mineral nutrients that could be possible to gain. So dirt would be a mix of the mineral nutrients. Salts and solids are held here. If you've got soluble materials moving past these mineral elements, organisms can do pick them up and store them or on the surfaces of the sand, silts, and clays. When you look at dirt, these materials are not plant available. So not something we really want to have in our soils, because that's not going to be growing our organisms very rapidly. If we look at what soil is, it's the mineral component, just like mineral nutrients, but now organic matter has to be added in. And that means you've got organisms doing their jobs because they've got the food to feed them. Different kinds of organic materials is going to select for different kinds of organisms. And really what you need to get good nutrient cycling going is what I just finished talking about. We need to have the predators in order to produce the soluble nutrients that makes everything go faster and better. So there's your source of all the nutrients that have to be in your plant so that the plant can grow as rapidly as possible. We've looked at some of these things, and we've noticed that you really want to have the biology in this system. So next slide, please. I'm sure all of you have probably seen this. This is the percent clay, the percent silt, and the percent sand in your, well, it's not, it's not, ah, it's an inorganic, it's an inorganic chemical situation. You've got to get the organisms in here. You've got to get the different kinds of foods that the organisms need to have. If this is what you're calling soil, and there are a lot of the older books that do just out and out call this soil, that's not right. These are soil textures. You have to add the biology, and you better have the foods for them to eat, or you will never build aggregates that will be able to hold together the structure of the soil and keep your organisms well fed. So soil texture is a whole different thing from what soil is. Next slide, please. So is it true that we need to know the exact process and the exact set of organisms who do the work in soil? And everything I've been talking about here is a part of what organisms do, the work that these microorganisms do in the soil. When we're doing scientific studies, yes, we have to know the exact process because we've got to be moving the knowledge gained, and the sooner we can really truly know what's going in that soil, the sooner it's likely that we won't have to wipe human life on this planet off. So we need to know how functions are performed when we're trying to figure out exactly how things work. But we know that the Green Revolution failed because the organisms who did the work were being killed by the practices recommended by the Green Revolution. That to me has always just been, guys, pay attention. Look at what you're doing to soil. You're converting it back into dirt. And that's not going to allow any human being to grow on this planet if we don't fix this problem and fix it now. The organisms that do the work are killed by the chemical practices. So any inorganic fertilizer, when it gets up to a high enough salt concentration, typically, is going to be killing a significant part of the organisms present in those organic materials. When you think about pesticides and problem organisms, again, those practices are occurring because of the chemistry that is being placed on these soils. And so here again, here we go with when you put on a certain type and amount of these inorganic fertilizers, the pesticides, the nematicides, all these toxic chemicals being put on the soil, you're going to be killing 50%, more or less 50%, sometimes as much as 80% of the organisms in the soil are going to be killed because of the addition of these Green Revolution materials. Why didn't we look to see what the effect of the soil organisms was going to be? Who's making money? Who's making sure to get their slice of the pie without any effort at being truthful about the long-term effect? We've now been using these kinds of toxic materials on our soils, ever expanding damage because we keep having to put more and more and more of the soil under agriculture. And that means we're destroying what allows us to stay on this planet, are being killed by the very practices recommended by the Green Revolution. We know that the entire soil food web is required for the plants to be healthy. So we need to go back to the bulge, if you will, go to the place where we still have those organisms that we can manage, get that back out on the land, and that's probably what I'm going to be talking about here in just a few minutes. Next slide. I think we should be coming, yep. We need to be monitoring for the measures of health. I'd love to be able to go through all of these interactions. So monitoring health, beneficial organisms increase and they protect the surfaces. Take a look at a healthy plant and the leaf material, the stem material, the flowers, the all parts of that plant. If it's truly healthy, it's got all of these beneficial organisms, so bacteria and fungi and protozoa and nematodes on those surfaces. And you can actually take pictures of that layer or two of that liquefied organic material. Compost is another name for it. When we make certain that we understand what the percents of high nitrogen, normal nitrogen, and woody materials, and we bring the temperature up in that composting process such that we have all of the beneficial organisms are growing rapidly, increasing their biomass. All bacteria and fungi and protozoa and nematodes, microarthropods, all of those guys. And the conditions in that compost is such that it will go in and destroy the bad guys. We want to make certain we get the temperatures in that compost up to at least 131 degrees Fahrenheit for 3 days, 72 hours. The first time we turn the pile in, so all the cold material that was on the outside of your compost pile or windrow, all of that material goes to the center, and what was in the center is now dispersed towards the outside. You've got to get that temperature up to at least 131 for at least 3 days. And then you repeat that. So all parts of that pile have been treated to kill the disease causers, the E. coli, the salmonella, the shigella, pastorella, in most cases, as we teach people how to do this. The amount of any problem organism usually disappears because we're over and over again heating that compost pile up and killing the factors that we don't want. We want to balance the concentration of fungi to bacteria. Well, both of those things have quite a different set of organisms when we're growing mustard, or when we're growing tomatoes, or when we're growing blueberries, or whatever you want to be growing. And so we understand how to manage so the bad guys are gone because we choose to be using aerobic bacteria and aerobic fungi and dealing with the problem very rapidly. We can go from start to finish on our compost piles in 28 days. This is not going to be something that we have to deal with for the next 6 months. Not at all. 1 month is all you need. And as long as you've got a decent kind of turner, that's really pretty much all you need. So we can monitor the amount of compaction in your soil by just taking a metal bar and start to push on it. And that shows you where the compaction has gotten so bad that you couldn't possibly grow those organisms. Where you see puddling on surfaces, we also want to test the compaction level, because we know then where we have problems, and we need to get in there and let the bacteria and fungi build their aggregates that allows oxygen and water and the organisms and your roots through those previously anaerobic areas. So now we can get well-structured areas of your field as rapidly as possible. What we find also with water is we reduce your water requirement in your fields by 70%. We can reduce that demand for water, and the water that gets sent down the mountainside down into the wherever it is it's going, and something that's drinkable until it hits the waste down there in the cities or something. But we've got to get this same attitude about water so we don't ever have requirements. Water should be automatically something that we deal with the problems. So watering requirements, reducing, we've done these experiments, we've done this work in Australia and some of Southeast Asia, certainly in South Africa and South America, where we've reduced the watering demand, and you can grow a whole lot more food if you don't have to throw away dirty water. Clean it up. Doesn't take long, but you do have to know what you're doing. Yields, always a good one to yield, to measure tillers. How many times did you have to go across your field in order to break up all of that concrete, which has normally been called a soil? What are the organisms inside something that you've got to apply tillers? What's that energy level? And it's just, you don't want to think about the cost. And yet we have a method for dealing with it it's right there if you would just stop killing the organisms. Plant color, another one. What's the color? You can use the color of the plant and look at how healthy it is. Bricks, it's not a 100% on the spot, but it is an in the field test that you can apply right then and there and have a pretty good idea of whether things are better or worse today as compared to last week. We want to deal with diseases and pests by making sure, I was starting to talk about this at the beginning, but I got waylaid. If this is our surface, top and bottom of the leaf, for example, and we have sprayed our leaves with this biology, you can see that there's a layer of really good biology on the surface of all parts of that plant. When we look at some of the above ground diseases, if you can get those leaf surfaces covered, the stems covered, all of the reproductive material there, we typically have very little, less than 10% disease of any kind. So really important to have those kinds of materials and know when to apply them, how much to apply. We are working with people who are doing drones so that we can just put like 42 gallons of a really good compost extract or a compost tea, whichever one you think is doing a better job. And you just drive that up and down the rows, spraying out the materials to cover so that none of those diseases and pests can cause a problem. Fertility, it's like all of this is all about fertility. So the soil food web, if you get the balance of the organisms, fungal to bacterial biomass ratio, if you can get that balanced, you don't get any weeds, or such a low number that most people say, yeah, there's a dandelion or two out there, but it's not going to cause me a problem. Now, if you said the other way around, yeah, your neighbors would not be happy with you. And then sequestered carbon. And I mentioned that just a little bit ago. And so I'm going to keep going. Oh, and I can see that. One more, heavy metals of any kind. So that we've done a lot of that kind of work. So getting bacteria and fungi are just amazing at breaking off materials. So something that was once toxic is now no longer toxic. So next time I'll have to focus in on that work. This is another, one of those things that you were told when you were a child that, inorganic fertilizers didn't hurt anything. And so we wanted to look at this. And the premise that I'm working against here is if you cut the top of the root system, I'm not saying that quite right. If you cut, and I need to see this, that if you cut, that finally connected. If you cut the grass, so we're using grass as the material to test this material. If you cut the top, then that caused the roots to fall off. So I kind of, as a microbiologist, that's just unfathomable that any good organic organism would just dump a load of organic material, regardless of what was going on with the parent plant. So to try to get this understanding through to people, July 15, a few years ago, Hendrika Schraven, the person I was doing this work with, and this is Hendrika Schraven here. He planted using ryegrass seeds, and then treated a part of the plot with inorganic mineral nutrients, pesticides, and other toxic chemical. Yeah, this is the common, here you are, this is Green Revolution. And so the expectation was that the plants in the chemical treatment would lack a deep root system, only got to 1 to 3 centimeter deep. Organic amendments. So in the same field, we applied organic material, and when we finished up, we cut the roots. And so let me show you what that is here. So Hendrika Schraven planted the grass seeds. Here we are on November 6th, where a week earlier, we had cut all of the grass down to approximately 3 centimeters height. To think of a herd of cows feeding on this. That's what they were trying to understand. And in the organic amendments, they didn't put any inorganic on, nothing at all like that. And we did look at the ratios of fungi to bacteria to indicate the health of these plants. So 7 days later, after we cut the grass, we took a PVC pipe that went down about 8 feet and had to pound that into the soil. It's kind of a lot to get. With the material in that, we had to take off the first foot and remove the pipe off. Do the same thing with the second foot, third foot, all the way down to the 8th foot. And as we went, we washed off the root system. So you can see the root system, really high concentration in there. And Hendricus is approximately here at right about there is 6 feet. So it's a pretty easy way to measure how far down these roots actually did. So if we listen to the average person who's not been taught much about root systems, this was well beyond what any of the growers in the area could even imagine. So they all borrowed the PVC pipe and went out and started doing this same sort of thing in their soils. And what they discovered was if you had that kind of treatment that we were showing, the standard chemical treatment, you would only end up with 2 to 3 centimeters depth of the root system. If we looked at the organic amendments, those roots were growing just fine, not going to have any problem. They're going to survive winter easily. So that come springtime, you're ahead of every other chemical trainer to show what was really going on. Hope that wasn't too destructured, sorry. Next slide, please. So we've been through the soil food web. So sunlight, energy, giving the energy to bacteria and fungi who get eaten by this next set, which gets eaten by the next set, which who is the top of the food chain? It's us. And so we are very much require looking at this material to try to make the connections in food so that we can all get through the coming period. Okay, next slide. I'm going to breeze through these pretty much, but soil microbe interactions make nutrients available to plants. We don't need fertilizers. What we're doing here is growing the plants, getting the mineral nutrients that you have to have in order to grow safely. You don't need to have fertilizers of any kind. The system will take care of itself. How does it happen? Well, predator-prey interactions, that was one group that we looked at. Mycorrhizal colonization, where the plant puts out the food resources, attracts the mycorrhizal fungi to that place. So the fungus grows a limb out and is now looking for where that food came from. And the mycorrhizal fungus is told by the plant that the plant needs X, Y, or Z. And so the fungus says, oh, no problem. I passed a lot of that this morning when I was growing.
Dr. Elaine, your microphone went out.
Can you hear me now?
Yes.
Now you can? Great. So mycorrhizal colonization, the fungus is going to go out and get nutrients for the plant, bring them back to the plant, hand over, and then the plant will supply the fungus with more foods. And so back and forth, back and forth. It's not a steady stream of one nutrient. It's a whole bunch going through the whole system. Okay, next slide. Flavor is determined by nutrient balances. Where does your plant get nutrients from? It gets it from the interactions of the biology in the soil. You just can't have something full of flavor if you don't have the microorganisms growing in your soil. So, the tomato of who's going to win the tomato and find out what tastes best, it's going to be those places where flavor was definitely being determined. And they go back, and it's the nutrient balances that are causing this. Nutrients are captured and held in the soil. So remember, I was talking about water use and how we hold and keep the soil right there at the bottom of the evaporation layer. All those microorganisms will be storing water in all of the root systems. So nutrients will be captured and held in that soil along with our water. So there is no leaching and there is no runoff except for maybe some water, but we're trying to get rid of that problem. Let's get all of our water going down into the soil below the plant. So you don't have to pay for water in some parts of the world. You don't lose any of these organisms. The root system of the plant is being helped by the interaction of the bacteria and fungi. And so by incorporation of that organic matter, you're holding those nutrients and the water in your soil. So you don't need to be buying water. You don't need to be buying inorganic fertilizers. You don't have to worry about some of the federal government comes along and says you're letting too much leach into your rivers and lakes and streams. You have to stop that. And it's easily stopped if we're paying attention to the biology in the soil. Next slide. So we just suppress diseases. We have our organisms sprayed on all of the leaves and the soil and the root system and every part of the plant that you can possibly think of. And that means we've got competition for foods. We've got inhibition because this guy's going to eat that guy's, and it's going to be consumption of pesticides. Suppress weeds by balancing nitrate and ammonium, so you don't have to be using herbicides. When you look at early succession of plant species, those are the places where we mostly have weed problems because things are very out of balance. You go through something with the big tillage equipment and you realize that it's ripped everything apart. How long could a human being and their family live in a place that is constantly having their homes and workplaces, everything wiped out by events? Same thing with weeds. We want to make sure that the bad guys come along and take care of them, balancing nitrate and ammonium. We want to decompose toxins using enzymes made by the microorganisms. We've got a little bit of picking and choosing that we have to do, but we already know from written studies, we already know that we can deal with toxins by using enzymes made by the microorganisms. Next slide. Building soil structure. So microaggregates are built by the bacteria. Macroaggregates are built by fungi. And they form pores and pathways that are great places for the microorganisms to live. Condominium houses along the edge of the river. The roots grow in these passageways, and they You don't need to be worrying about water. You don't need to be worrying about diseases, all of those sorts of things. And then sequestering carbon. We certainly have sequestered more carbon than anybody else. In small trials, we want to take that to larger trials so that we can take all of that waste food material, make certain people understand that we're sequestering carbon, and we've got to mix that properly with soil structure so it will be a benefit, as well as sequestering all that carbon. Next slides. So looking at the soil biology, bacteria, well, what this is like slices out of various parts of this, going from a place where you have no organisms growing at all, a little bit, more, and fungi finally come along and start doing their thing. So now bigger plants can grow. We're changing and altering the bacteria and fungi are being changed, and fungi are being changed species and diversity things, and so different plants grow. As those different plants grow, now there are different food resources for the microorganisms to start a whole different type, totally different community structure, just like what we see with the trees. And a little bit further on, it's changed enough so that these plants are growing instead of those plants. And you build that structure, and I always notice that it's, these are microorganisms. That's the only thing way out here in a highly disturbed area. And Mother Nature will slowly but surely build that structure so that we finally get down to the highest structure of materials, the old growth forest trees. So understanding that movement from 10 micrograms of bacteria to no fungi at all here. 100 micrograms of bacterial biomass, only 10, but it's more than what we had before. Keep growing and keep moving forward, balanced, not quite, yeah, this is a little bit off because of the transferring it this morning. So balanced would actually be the 600 micrograms of bacterial biomass and 600 micrograms of fungi. Well, then it's moving now into the fungal dominated world. There's more fungal biomass in that soil per gram than there is of bacteria. And then bacteria again, kind of hanging out at one level, but the fungi keep growing. They keep increasing. We have started to work with people like Paul Stamets, for example, different folks here in Washington State, where we're looking at, we can get the fungal biomass here up to not just 7,000, but 70,000 micrograms of fungal biomass per 700 micrograms of the fungi, of the bacteria. So when you think about what's being sequestered here, massive sequestration process going on here. So next slide. It's like, where do you want to put your plant in here? If these are places that are going to select for blueberries or blackberries or tomatoes or kelp or whatever you wanted to grow, wouldn't that be your first thought, that I'm going to go and make my whole field? I'm going to make sure it's got the fungal to bacterial biomass ratio balanced properly. I want it to make sure that we have all of the proper balance to support these plants that we want. So next. Yep, so let's let Mother Nature use let us understand Mother Nature's use, and I want to start looking at uh, where what does this term bacteria and fungi, protozoa and nematodes, microarthropods, things like that. So how about if we do a little bit of showing those organisms? And I need to I need help. So I need to change to my next pair with a zoom. No, I didn't practice this, so I don't know how to move on to the bacterial, the biomass. Shadow.
Do you need the other slide up?
Yeah, that's what I'm. Aha, now I remember. This is what happens to me all the time. Small price for being alive at 71. Yeah, so
I'll stop sharing and share the other one.
okay. There should actually be two other ones. I don't want the one that has
You want the microscope?
I want the microscope. Thank you. All right. So looking, is there anything that anybody wants to, burning question out there that I can answer real quick? Well, I'll leave you to thinking about it. So use of compo microscopes to identify organisms in whatever you want to put down on the cover slip. That's what we're going to be doing in the next part of this training course. So next slide, please. This is a suggested microscope. You have to kind of stay alive, working with microscopes to know what the best and the least expensive. Of all of the different microscopes around, this one's been around probably for the longest amount of time. When you decide that you want to be looking at the microorganisms, I recommend that you go and find one of our, get hold of one of our the people that we've trained, so that this part of the system isn't too difficult. Or maybe there's a person at the high school or college close to you, and you can get their help. Binocular means you've got two eyeballs, and so therefore two places to be looking down your compound microscope. All of the other things that are required here. Next slide. So this, you can see that this is binocular. There's place for both of your eyes. So there are lenses in here that you can look through. That when you look through it, you'll see about a 10-fold increase in the size of the organisms. So you usually put your slide here that you've prepared, and then you dial in the objective that you want to use. Typically, the 4x objective is good for looking at some of the bigger things. Over there's a 10x eyepiece there. So you've increased the size by 10-fold. And then there is a 40 X magnification, and the 40x plus the 10x will give you 400x. Most of the time, we have to use the high magnification in order to see the bacteria and be able to count them. Fungi typically the same way. Fungi like to kind of hide themselves in the material that has to get pulled apart so you can see a little bit better what you have in that sample. The flagellates and though the protozoa, the nematodes, typically 400x to 100x magnifications. So you put your slide on here and take a look through here, and I'm going to be showing you a lot of what the organisms look like. It takes a little bit longer to go through all of the why did you do this way versus do that way. So that's something that you'll come to the Soil Food Web School and be taught by people that every time you have a free hour worth of free to you, you'll hop on your microscope, get hold of your mentor, and be able to get some of the reasons why we do things in a certain way. So these are, you dial in higher or lower by moving this handle. This handle, there's one on the other side as well as over here, so it makes it really easy for those of you who, some people are left-handed, some people are right-handed. I do both, just to confuse everybody. This is the stage, that's the name of this part, and the stage has lenses in it so that it can focus right on your sample. So the next thing is the light at the bottom. You need to have something that you can make certain is aligned with this lens here. So other than that, I think we've covered it. Next slide, please. There we go. Eyepieces up there, objective lenses, just to give you some of the jargon I didn't use. There's your stage. I did mention that. This is called the condenser, where you're focusing the light coming out of the where the light comes out of, and comes up and through, comes up and through. So you want to do a little bit of turning down, tightening up when you first get on after a day's worth of work or somebody else has used the microscope. And you want to do a little checking to make sure that all of the organisms on this cover slip and on the slide are already nice and focused. So that takes us all the way down to there. Brightness control. You'll notice as you go down through deeper and deeper, the brightness gets less and less. So the brightness is down here. That's what you're going to adjust. That's your dial for adjusting that, the brightness level. Coarse focus, and you'll go up to fine focus, so you'll learn the ins and outs of that. Condenser focus knob, that is right there for getting the edges of that of your condenser just focused on the best you possibly can. Now you're ready to go. Next slide, please. Iris diaphragm. Oops, one more thing. you can tighten it way down to the point where you get just two bacteria in your field of view, or you open it back up using that iris diaphragm, and the control is that knob I showed you on the other side. Stage controls, so this pushes your stage back and forth. This will then give you the left and right. Yeah, one goes this way, one goes that way. So where's your sample? And when we want to count multiple fields, we'll go right across there using this microscope. Next slide. We always do multiple assessments. So if we've got a sample, we're going to go at least 3 and maybe as high as fungi. Sorry, as 4 or 5, just because we want to have some understanding of the variability of these organisms in our sample. If you look at your first core and it's 3, and then the next one is 6, and then the next one you look at, you're seeing 7. Things are all tightly bunched together. I wouldn't worry too much. You really wouldn't have to go on to the next field and the next field and the next field. So you are now done with that sample for looking at bacteria. You would go on for fungi, do the same thing. So let's say with the first fungal strand you look at, that's about 1.2 micrometers in length. It's about this diameter width, so of 4.0 width to the slide to the fungal hypha. And then when we go to the next field, randomly, you just have to close your eyes and move around and look. Okay, there's things in this material, so I'm going to sit and count this one. And so another fungal hypha, this one's down at 0.02. And you go, darn it, I'm going to have to do more fields of view because there's too much distance between 1.2 fungal hyphal strand and a 0.02. Okay, so you go to another field and now it's got, it's way out in left field. So what you want to do there is then clean up your slide, mix your sample better. Make certain that you take that sample so that you're more likely to have a uniform set of fungal hyphae on that field that you're looking at. So showing you here, how we go through that process of making certain that we're getting as the same spread all through your samples. Next slide.
Dr. Elaine?
Yep.
We're coming up nearing the ending of our time. I was wondering, instead of going through more slides, is it possible we could stop now and ask some questions?
Sure. How about if we do at least one bacterial slide?
Okay.
I'll take a few. Yeah. If you've already lost it, that's okay.
Well, I've closed it. I just think at this point, our audience is going to start leaving because we usually schedule for 90 minutes. And I wanted to give them an opportunity to ask you some questions. So, Chris, if you have something, if you want to start the questions, please.
Yeah, absolutely. So I'm going to combine a couple of questions together here. We have quite a few questions. So someone wants to know if you can recommend soil microbe products. And then a similar question is that I'm limited to 5-gallon pails for my garden. for my vegetable garden. Can you suggest a good bagged soil product?
I'm going to go backwards on the question. Yeah, what you want to do is find out who's started to put the food web on their label. So it should tell you the active bacteria, the total bacteria, active fungi, total fungi, protozoa, nematodes, good guys or bad guys. So that, if the values are above the and they typically have the desired range that they've put on the compost that you're buying from them. They'll ask you typically, what do you want to grow? And so they'll put something on on the bill or on your whatever you're going to hand them to be that they can take that home with them as a little booklet, for example. And you want to then do some of the microscopy, so you can say the bacteria were doing much better than the required minimum. The fungi way above the minimum for having a good compost. You had really good protozoa numbers. You had really good nematode numbers. And so you could then compare and know if that person's compost is going to do the trick for you. And then the question becomes, how much do I have to add? So if you look at the microscope and look at the amount of bacteria and fungi, protozoa and nematodes, and what's that balance? And we go through all of this at the school. And, that's what we train you on. So you can go ahead and start helping other people. Or if you can only get compost that's somebody else's built, it'll tell you every time whether this is what you want to be using. And typically then, when you know what is in that compost, you know what the amount is that you are working on, you can do the proper balancing. And this is where it might be easy to get one of our one of the people who's always online and will chat back and forth with you on the how well that's working out.
So Dr. Elaine, does companion planting help with soil health?
Yes, it does, because it's going to promote the diversity within that system. Think about the fact that all of these things may have been treated with herbicides, pesticides, all kinds of things. When you bought it from the store, for example, and there you want to start spraying that plant with the beneficial microorganisms that will cover the surface of all parts of that plant, so that you're killing off everything. And finally, we get to do the good stuff, the good kind of killing, to get rid of the E. coli, the salmonella, the shigella, to get rid of the verticillium, all the different things. The root-feeding nematodes will be killed by these organisms as well. So the beneficial organisms are what you really want. And it's probably better if you make that yourself and then go and buy things from the store, because that will we help people understand why that's worthy.
Okay, thank you. So how can we increase compost in a bin to 131 degrees?
You typically don't have to worry about that, because we've already figured out the ratios for the different kinds of plants or different kinds of compost that you're going to be putting around the plants. So generally, we're looking for woody materials, that will be 60% of your compost. And we want you to vary those woody materials. So as much as you possibly can in that 60% of woody material. Then you've got 30% of green plant material. And so same thing there. You want to make certain that you're 30% of your pile. And you do not have to be crazy about, it's not 30.100000000. No, you would drive me crazy. It's kind of more or less in that area. Because with all of that different kind of plant material, there's lots of variation in the fungal to bacterial biomass. It's, some things have a handful of turkey meat left. Well, over here you got sugar, and over here you've got, so don't worry too much. You're going to mix this thing over and over and over. So make sure you've got the 30% green, and those are anything that was harvested and still is green. If you let that green plant material dry down really fast, you can hold all the nitrogen you want in that plant material. But if you let that decompose really slowly, and it's real wet and it's real sludgy, kind of ooky stuff, you will return that to the woody pile and get that for the next time you build a compost pile. Okay, so now we've got 60 plus 30. We need 10% more, and that's high nitrogen material. So things like any of the clover. Dig down, find and make sure that you have nodules on the root system of those green material, the clover. There's a number of different things that the nitrogen fixing bacteria will produce extra. So you look up information on that particular set of bacteria and make sure that their nodules are present on the root systems of the grasses or the all the different things that can become hosts for the plant. So now you've got 10% high nitrogen, 30% green, and 60% woody. And you want to just mix that up in the container. We generally have hard times getting really good temperature being in the composting that you're making. So you want at least 3 feet wide, 3 feet long, and 3 feet high. That's the minimum. If you get more than that, great. It's just going to be less work for you to get that compost to start doing its thing.
Thank you, Dr. Elaine. That was a great answer to that question. Next question is, does electromagnetism play a part in soil health?
It probably does, but we're not very good at the cost of making that equipment. So I'd need to look at the kinds of machinery that they're using. How are they putting the light on the surface? And a lot of times, correct me if I'm wrong, but a lot of times the surface of your plant is going to have a layer of all these great, wonderful microorganisms. And the thicker that layer is, the less you'll be able to do anything with the electromagnetism. And I don't know too many people who want to sit and wash their leaves in order to be able to to do that kind of work. So I'm not right up to the last minute in my understanding. So, it's sort of like, yeah, let's start a conversation, and the person who asked you could go ahead and give me a buzz on my internet, and I'll do my best.
Thank you so much. Okay, so there's a series of questions on manure. So there's a question about aged cow manure and how that compares to horse manure and rabbit manure, and its relation in usage to make the soil healthier.
Yeah, it's knowing what has happened previously in the life of these organic materials can be really difficult. You don't know whether like with the horse manure, people go out and give the horses doses of toxic material to try to kill everything that's going on in the digestive system and not allow any of the pests or all of the other disease causing it all. What you have to talk to the veterinarian usually, because the most people will not have a good idea of what exactly was applied to their field or to their animals. So they're all, you gotta be around those people with that might want to give you some horse manure, give you some pig manure or something. It's routine. If people have a large number of horses in their barns, they are going to be feeding those horses with some pretty toxic materials, because they do pick up some really repugnant material, smells and stuff. And because then also that manure is no longer high nitrogen. People use manure, cow manure, probably pretty much okay, as long as it hasn't been out in the field, frying away in the sunshine. If it's something that was, the animals are brought into the barn to be milked, and in that time period they're pooping and peeing and all of those wonderful things, and that's a good time to go in and pick up, where you can see exactly where the poop was and where the other stuff was, and take the manure is what you really want as a type of high nitrogen material. That's what really gives you temperature in a compost pile, is the bacteria and fungi start growing inside of that pile, just having a grand old thing, and the temperature just takes off. And you can reach 150, 170 degrees, no problem. And sometimes if you haven't been real careful about getting the percentages at least close to each other, you'll overdo on the high nitrogen, and you got to turn that pile then. You got to deal with it ASAP, or you may no longer have a house, because it gets that hot. It doesn't take much more than a piece of paper to fly through, or so there you have to be pretty, pay no attention to the temperature. And we really expect that you will have a file that says each day what was the temperature of the pile. So you can always go back and what food resource gave us the best result. Well, those horses must be sprayed with their horrible disease something or other, because we can't make good compost when we use theirs. So we'll go over here where that's a organ or it's they don't feed those animals anything they shouldn't be fed. So keep your eyes open on that. And people will make will use beer mash for the high nitrogen containing part of the compost, and that usually works out very well.
Well, Dr. Elaine, I want to thank you so much. We've come to the end of our time. Everybody's been posting in the chat just how much they appreciate all the information that you've shared with us. And I want to thank you once again on behalf of Connecting Consciousness Worldwide Voices for joining us today. Certain people are going to have to watch the video just to get all the information again, because there was just so much, just jam-packed it full of information. Again, thank you for being here, and thank you everybody for joining us.
Thank you very much. It was fun.